2 years later.
A warm welcome to everyone present here. Today we have gathered at day 4 of the GIAN session on enzymatic protein hydrolysates and material behavior of extractions in human nutrition. Today we will be having 2 lectures, lecture 7 and lecture 8. Then after lunch we have tutorial based on the above 2 lectures. Today we have Dr. Pooja Gulati with us, who will be the faculty in charge for today. Now please take
Good morning, all of you. Hope you are enjoying the sessions. So we'll resume with the day 4 today. I'll be coordinating the session. If you have any queries as we are doing the discussion, so we'll continue with the discussions, and any problems or any suggestions will be most welcome. So let's start with the session. Please come and thanks.
Good morning, everybody. How are we? Okay. The day is very beautiful outside, so must be enjoying the sun and also enjoying. Let's start our session about day 4, session lecture number 7. We'll be talking about some flavor release here, and then last is impact, particularly in metal science perspective, and thermal properties, the glass transition temperature, and the protein. Why we study material science and combine with the physical and chemical and functional properties, because they are correlated. So definitely we have to have some answers and questions. The science challenge, or key challenges in this area, relating protein molecular properties and microstructure to the functional behavior in low water condition. Particularly in ingredient manufacturing industry, all of the sprayed out powder for the formulations, whether it's pharmaceutical, food, or anything, you see the range of powder will be around 5 to 6 and maximum 7% moisture. So that's low moisture. So the free flowing candidates and then the functional properties depend on those. So if you then the water uptake or addition of more moisture, the glass will be there. And significantly, when there is a glass transition temperature occurs a certain percentage moisture, and then most of the higher content, there is a glass to rubbery transition taking place. So the glassy behavior is now the rubbery material. So we'll be discussing about the material science perspective, how this physical, chemical, and functional properties that are affecting all this condition in the vicinity of glass transition temperature. So another size challenge is that predicting and testing improved novel properties of pure protein planes and formulations. Food is a complex molecule, so even an assembled food product is no longer the protein itself. You have starch, fats, fiber, and other properties, other ingredients. Protein will be inherently interacting with these other physical barriers going to transportation across these things. So according to that, mechanical behavior and functional property will be determined the ingredient composition and how they interact with each other. Like, I gave an example, if you have gelation properties control, like tofu, I gave an example, was it 11s or even 7s, the functionality is lost because they were disulfide bleach. So what is the design rules for the protein? So you have to have a simpler model first, understand one protein, then challenge the environmental condition, the pH, temperature, pressure, and any other parameters that we have been discussing. That's called the microenvironment. So we understand the system we are working on. Even an enzyme challenge protein when hydrolysis being produced. So we know what the optimum condition we have and what are the enzyme concentration, what pH, what temperature, because these have determined the effect. If we know the end product, so we have a desired function. So repetitiveness will be producing the same over a process time. So we have control of the reaction mechanism. So formulation is basically based on understanding the interaction of these molecules And then everything has to undergo a processing condition. In a pilot plant, so you are doing a pilot plant like if you have a high pressure processing. So this molecule has to be subjected to high pressure. Under high pressure, the denaturation will occur. So this is an example. So when you go to industrial scale, so same processing condition is happening, temperature, pressure, shear. So you have to have all these things parameter known. The basic example of simulated extruded product is capillary rheometer. So if you have controlled shear, pressure, and temperature, the product extruded come out. Like the complex that we use is basically an extruded product. So if it's hydroscopic, so the crackling sound. Recently, one article came, I think the last month one article came on Science from a group of Australian researchers published in Science of this molecule collapse when you put in drop of nail. So disintegrating and all. So they come up with this article. This is the front-page cover page article in Science. So the processing, storage, and desired properties. So all these conditions will be like I was talking about the aging phenomenon. Then how we store this product. Storage, as we mentioned, like hold this at 7 degrees. You might have read in some of the specifications written in your MSDS. Please store this at refrigerated condition. Store at -70, -20. All this because storage condition is very important. So desired property is to have that desired property storage condition is important. So glass condition temperature will be telling that way. So what is actually the material property? Material means the raw material, basically. Raw material can be protein, starch, sugar, any of the things. So that's the material properties. How these ingredients, these molecules behave on those conditions. So the key to food manufacturing industry, like we have desired functionality, regulars texture, or the mouthfeel, the organoleptic property, the sensory attribute, because these are the key drivers from the consumer perspective. The function, the product, you have designed the product, but the consumer, the palatability, the organoleptic product properties are not liked by the consumers, the end users are the consumers, and the product is spoiled because we have not studied, not assembled the molecules correctly. Maybe somewhere, if it is off flavor or is bland, so they are not accepted. The food is just like sometimes we put in mouth, immediately we say, oh, food is good, then we take next food. If it is not good, we say, okay, I can't eat it. So you can understand. Consumer, same thing, the consumer process is driven like that. They will try the food once, second time they will reject it. So we have selected certain conditions like moisture. We discussed about water molecule, water creating structure, water inhibition, plasticizer. And in any formulation of food, we have water in that. Even though you are in middle, we discussed about 87% water. So water molecule is a plasticizer, so how water behaves structurally. Then different operation series. It's a large series like anions and cations, chloride, bromide, iodide, and cyanide. So in that series, we are studying about how the salts affect the protein conformation in the solution state. So then you can have the salt interaction. And so this way we can have simpler model fast. If pure protein, so to understand the kinetics and all the behavior, material behavior, then move to simpler to complex. Then we can assemble the product. Then pH and polymers. Polymers is basically sugar. So coming to pH, it can be acidic pH or basic pH. You will see the pH. We discuss about the solubility of the barrels of copper sold, solubility that here the isolated point. The solubility increases away from the isolated point. So you have to have like pH effect. And if you have acidic, strong acidic and strong alkaline, we have already discussed that denaturation taking place away from the pI, net charges reduces. The protein no longer in the negative configuration. So that's why the structural hierarchy is different. We discuss different structural hierarchies. Alcohol is turned into a random coil. More stable structure is now protein now exposed to outside. Effect of sugars, and then nowadays processing condition is such a people say how much sugar in that. Sugar may be simple sugar or complex sugar. So these are the effect of that. So then effect of temperature. We already have discussed some of the classical examples we gave how the denaturation temperature is occurring. Means you process the food, that's why it's overcooked. So this means you have denatured it. Then I discuss some of the references here, like when you have denatured protein, it's no longer a native protein. So denatured protein may not have the same biological function while we are looking at a native protein. You are paying some protein is completely denatured. You are buying some of the soy protein isolate or whey protein and completely undergone denaturation. Their functionality may not be the same with the native protein. Of course, the nutritional property will be different. So understanding protein, the effect of temperature, then what is the optimum temperature? Every protein has to expose to the temperature because in a real condition, so all this plays together. In a function of time, temperature versus time. Enzyme kinetics we study, like if I am cooking something, then how long we cook it. So give me an example, how long we keep it, how long we cook it. So I give you the yogurt fermentation process. We say that heat the milk for half an hour at 85 degrees centigrade. Why? We are just unfolding the protein and denaturing the gas molecules so that random interaction takes place. You give a strong gel where you have weight determining weighing off of the synthesis, preventing the synthesis. So there is not much water when there is complete fermentation taking place. So that way, so how long we have to completely denature the temperature. Suppose I say heat at 100 degrees for 2 hours. Maybe you have a charred flavor, burnt flavor. It's a yogurt, but people will off flavor. It's a burnt flavor. So people will like clarified butter. So you will heat it above that 50 degree, 60 temperature to no longer have the same flavor. So that's the temperature and time. Then hydrolysis, particularly enzyme. We have been discussing different enzyme hydrolyzed production, how the time and temperature and all that. We have you might have a bit of idea now. You can teach this. So processing condition in real food permitting. Processing conditions in that means when you process something, the food, like you are going to extrude line or any. Just to give you an example here. Switch under temperature, pressure, shear rate, moisture. Something is a dough baking industry. I'm talking about. If it's a bread or bread in the baking industry, you know the starch to protein ratio, how hydration is affecting in terms of gluten network development, how differential binding of water is taking place, and how the starch network is. What kind of starch you have? Protein to starch is very important here. Then the gel network, the dough network will develop, then dough extensibility. So those are the processing conditions. Just give you an example. In confectionery industry, also manufacturing chocolate, anything. So it's a complex molecule, simply complex. So then why we are employing a range of instrument techniques here, different structures here? Why? This is very basic. Everyone can ask question. Can I not able to answer the question here just in simple way? The answer is emphatically no, because the molecules vibrate in different length scale. So I have shown 2 different slides how to integrate those, and then the complementary techniques. Suppose we are interested in studying the physical properties, and then we relate to the other parameters. If you are studying the glass transition temperature, protein, or any biomolecules like starch and all things, you have to use differential scaling calorimetry. I'll come to that later. But that's a complementary technique. This is routinely used across the globe. Then purification process managing chromatography, SPLC. That's for extrusion process. The real environment is that capillary electrophoresis. CR is the rheometer, capillary rheometer for extrusion process. Then capillary electrophoresis to check the purity of the compound. We discuss the bioanalyzer. Suppose you have a protein, you purify it, and you want to say that the protein is pure, so you use electrophoresis. That is a simple way of checking the purity. Then dynamic mechanical analyzer. This dynamic mechanical analyzer, DMA, it's basically and keep the storage modulus. That correlate can be DS experiment. So then we discuss why there is a spectroscopy like FTR and FTR. Like, if you have food system, you may have solid, semi-solid, solution, powder, or liquid. So different format. So different format means you have to design which instrument you are going to select here. FTR has versatility in that. It has different accessories. So if you are using solution, you can use calcium chloride indoor, or if you are using a zinc sulfide crystal, so you can put solution there. And if you are studying the heating system, you can hold the sample in the sample holder and insert the sample holder, then increase the temperature from the digit, goes to all the phases and monitor the spectral analysis. Then you can collect the spectra offline, you can analyze the data left. So the way you are looking is that you have the probing the structure, how it is happening, and that will determine what conditions you are interested and what you are looking. Then you design. Then I'll come to FTR. Yesterday we discussed about some in the tutorial from a spectroscopy. Basically, if you have some of the wet condition, like signal to noise ratio is very important here. Signal to noise ratio means that your sample is getting noise, so it will introduce an artifact. So you will be biased and selecting either a peak is coming from the artifact. So I showed you two comparative tables showing that even though you are having powder versus some wet palette, both are having giving similar ranges. Means you can study in a hydrated condition the sample other as a free flowing powder. So it's a versatility of that equipment. Also you can have accessories to study like gelation way. You can use a capillary, you can stew, otherwise you can use an MR tube to heat the sample in a heating block and then directly punch the NMR tube to stop the reaction and then put the NMR tube in the sample holder of drummer and then take the spectra. Then you get the structure of the gelation there. That is the technology has to be applied. So these are complemented. Then coming to circular diaphragm. We know some of the biologically active and functional molecules are very expensive to explore in that way. The amount of sample we need in FTR and Raman will be in grams quantity. And if we cannot afford to have to hold that kind of sample in those studies, we have a buffer system selected in circular diaphragm. Basically, it's on the chirality of the molecule, the promote force present in the protein, on left hand or the right hand, that determines the circular diaphragm. Maybe far UV spectra and the near UV spectra. The far UV spectra gives the second structure. Near UV gives the tertiary structure, how the tryptophan and all the amino acids are crowding the structure. So the advantage of circular diaphragm is that we are using less sample, but we should have a buffer system which is completely soluble in protein, no clouding, and the buffer system doesn't interfere with the chirality of the molecule. So each technique has some advantages, some disadvantages. But we cannot study insoluble protein aggregation and other products in CD. Circular diaphragm, it has not been applied that way, but recently people are trying to integrate some technology. So these are complementary side of technology, so we are using. Microstructure. What is microstructure? Suppose I give a thread. You are looking at my microscope. We are doing confocal laser scanning microscope. I have stained appropriately. Starch is stained differently. Protein is molecules stained differently. The fat molecules stained differently. And looking at the microscope, that gives you the microstructure of the bread molecule. Because when you're making a functional bread, which is multigrain bread or something to eat, you have to understand how these molecules behave on the GI tract. So you can do RT and GIAN outside, and then in vitro studies you can monitor the changes. Pepsin diet in the protein, alpha amylase digestion the starts. So microstructure is designed in such a way. I'm just giving an example here to look the like gelation, aggregation, and the other in the real food system. So it can be, we have discussed about scanning electron microscope and transmission electron microscope with few examples. Like scanning electron microscope, we discussed about the gelation of the globular proteins. Then transmission electron microscope is microaggregates, the high molecular weight protein has been in the scanning electron microscope. Confocal laser scanning electron microscope, I just discussed about. I'm not showing an image that. But moving to the structural characterization, we can do structure in FTI like alkylis, beta-acid, Roman can also tell whether it's random coil or not. But the rigidity of structure and the location of the water molecule here, C13 carbon NMR has been employed, and the relaxation NMR is there. But some of the other nuclear cell scattering also used is for improve the protein structure. Then coming to why we measure TG, why microenzyme, why we measure TG, we can ask the question ourselves. So the glass transition, what is TG then? Is the temperature of the material changes from the glass to more rubbery like liquid like stuff in our first food. Particularly that controls the material properties. You can add any of the plasticizers like whether it's water, sugar, or low molecular weight additives. Low molecular additives. I think we'll be discussing the hydrolysates that we produce. Some of the fragmented hydrolysates, it acts as a plasticizer. It means if you blend that with your native molecule, that will reduce the molecular weight, it's clearing the peptide form as plasticizing the TG decreasing down. Why it is happening, we'll discuss. So the idea behind it is to have some of the low molecular weight actions and then study the structure. It's a case study we're discussing here. So we discuss about different length scale. So you can see we have crystallography here. You can do by X-ray, or if you're going to vibrational spectroscopy and the neutron scattering microstructure from here. We all have seen polarized light microscopy. The birefringence comes in a starch molecule, and when it overheats, the birefringence disappears. In the circle comes staining procedure. That's a microstructure there. Means if you have amylose, pure amylose, the which starch, and just put in water and heat amylose constantly up to the gelation point, then you see when the amylose being heated, the birefringence disappear, means it's completely gelatinized. You no longer have the form, the molecule loses crystallinity. So that's what the microstructure says. So glass transition temperature is always measured on the second scan of AC. But to come here, and so I'll be going this slide and next slide, then we will be discussing how to measure the glass transition. Please interrupt me if there is any doubt in the middle of this session, because this is the really challenging session. We have to be clear to move next step. So anytime you can just stop me, okay? But we have ample time at the end to discuss in the tutorial. But so this slide means what is happening. I'm just showing some thermal events. So this, look at here. I was talking about powder, which is roughly 5 to 6%. You all know that the sample will be 7% moisture, no longer that in any powder. Then slowly the moisture content being increased. You can see that an inflection point is happening here. You can see that the slope here, inflection point. Okay, I put a straight line here, straight line, another straight line here, and middle inflection point. That's called PG. That's the heat capacity change. Then this is the second scan. I will come to the first scan and tell. I'm just telling about. And you can see this overshoot here is the denaturation temperature. Then how to measure it we discuss. We'll be discussing also in the afternoon in our tutorial. Then we will not confuse the practical aspect of the data here. So we'll have to measure. Then I will come back to this slide. Now, same moisture here, you are hitting from -20 up to 180, I think. You are hitting up to 180. You are slowly hitting 20 degree. I'll be discussing the parameters that hitting employed here. So, if you see this peak is called the aging peak or the stress relaxation peak. Means if your sample has been aged, that is any food item we buy the ingredient protein powder is little best before or use by that. If you store past that one, means say aged, 2 years of age, no longer have the same, how to monitor that? How do you know it's been stored for 2 years? We are buying a 2-year-old powder. So if you put in DSC and take a test powder as your control and monitor the same sample, a 2-year-old sample or 1-year-old sample, this relaxation fix, the Tmax, will be different for a test prepared sample and the aged sample. If it has been stored at the same temperature, same condition, okay, coming from the same factory. Then the glass transition temperature, you can see it has already been there, but we cannot see on the past scan. This is the past scan because due to the overlapping nature, so slope is not that big to integrate to get the glass transition temperature. This is the protein denaturation in the term, means the denaturation temperature, which is decreasing with increase in moisture. This is 26% here, 27. You can see the denaturation temperature here. It was 180 close to it. Table will be clarifying. So it's 130, 50 degree lower than the compared to the original one. Means water is plasticizing the molecule. The glass transition temperature is decreased. At the same time, the denaturation temperature also decreasing. Coming to here. When you do a first scan here, second scan here, you cannot see the, do you see the, notice, please carefully notice this one. You don't see this peak here in the second scan. This peak means the aging peak is not there because we have erased the peak. We heated that sample, the history, thermal history has been erased. So we freshly prepared a sample. If we hold this sample, erase the thermal history, just hitting over to 60, 70 degree, and now same sample we hold for 1 day, 2 days, 3 days, 5 days, 7 days, up to 1 week, I'm telling. And we monitor about the aging effect at the same temperature, just simple experiment, and then you see the aging is increasing. Means the sample is aging. So you can still study the effect of aging there. So the glass transition temperature has been measured. Good. Now we see the graph that with the increase in moisture, both TD and denaturation temperature and the glass transition decreases with increase in moisture. Means what is plastic or moisture. Now, coming to, we talk about application of infrared spectroscopy, studying the structural aspect of the molecule. In this, we all know that assignment of this, the table, one table is there, will come later. This amide 1 region is 16 to 17 and region is the region of our interest. Because the protein amide 1 region gives the structural information, there's amide 1, amide 2, amide 3, but there are many concerns about amide 1 here. We'll be discussing how to set up that experiment. I'm not going to that aspect. To take the original spectra of the native protein, then apply for the simple. If you look at the midpoint here, you can see this protein is predominantly beta C type of nature. All plant, mostly plant proteins are in this category. Some of the animal proteins, native structure is alpha helix. That's why I was asking, what is the native structure of protein? Mostly it's alpha helix, beta C, this kind of structures. Then what is the digital structure? It's the random coil structure. It's completely uncoiled and completely denatured structure. If you take a denatured protein, it may be antiparallel beta C, or it may be under complete denatured, it may be random coil. So the first graph shows the peak here, 1634, correspond to beta C type structure. So the proteins are proteins predominantly beta C here. So if you use an algorithm here, it comes along with the software, the parameters is there. You have to set within the parameters in the software to separate these buried peaks. So this is called Fourier deconvolution. What is Fourier deconvolution means? Resolve the buried peaks without affecting the spectral contour. You are not introducing any artifact. You are not introducing any peak inside the original peak, but you are resolving it, the other buried peak. You are separating peak from peak. So you can see the peak is now bit random coil talking the protein tag. Then the percentage, how much percentage beta C, how much percentage random coil. But dominantly the peak is here 1634 with the beta C followed by the coil and random coil. You can see the protein may not be 100% beta C. It may be 20%, 30% of that, another percent. So then the composition varies, so predominantly beta C. So that's the way the protein is being characterized. Yeah.
I just want to have some queries.
Yeah.
First of all, what is on y-axis? Is it adsorbance or transmittance?
This one is observance. So you can have transmittance as well also.
So how we in second case, like you have said the deconversion of the peaks. So this deconversion of the peaks has been generated from the same spectra, or we have generated some other spectra to identify these peaks.
Okay. Once you do the Western correction and you can subtract your control. You have control can be anything, and then you have a corrected spectra. The same spectra will undergo the deconvolution that you are using original spectra.
Basically, we simply do the when we are checking it, we put the scale of 1 to 100 on max.
Yeah.
So now we increase down to 1%.
Yeah.
So automatically these peaks will be visible.
Yeah.
So now there is one more thing, this because basically in FTI we got some vibrations and vibration, bending and stretching vibrations. So when we are doing bending and stretching vibration, we generally get some peak, and that peak corresponds to a particular band.
Yes.
It may be in carbon-carbon single band, primary, secondary, and tertiary. So basically, when we are saying that you have beta sheets, random coils, beta turn, so actually is it true representation of the structure using these FTI data?
Yes. Basically, protein is basically what? Carbon, nitrogen, and iron. So basically, those molecules. So this is a pure format you can assign to that. Yes.
Basically, what we are here proposing that it could be a possible structure, but we cannot validate in this particular matter.
No, it's validated. Or it's across the globe. People use either FTI.
Yeah, I agree that if you want to see that if you have two samples and you want to make a comparison, then it's good that you can identify which peaks are missing and which peaks have increased in terms of resonance and transmittance. And then you can map back having an understanding of what is the nature of the sample. So if you know the nature of the sample, then you try to predict that particular vibration because it's coming from the primary ring bond.
Yeah.
So if the primary ring bond is actually adsorbance is increasing that particular rate, so we can conclude that this particular sample, the primary ring group has been increased. Now, if we know that our sample is protein, then we say that amine groups has been increased. If our sample is some polymer, then we that the polymer has been increased or some amide bond has been formed. So, but these are simply mere speculations because you cannot truly tell what it is there. Because if you talk about the protein, I agree. We are talking about beta sheets, helix, and these things. But in addition to that, if we look for the amino acids, they have the hydrophobic, because they supply the long chain chain, aromatic chain, and these chain also give the vibrations. So though they are not a part of the structure of alpha from the beta sheets, in majority of the case, still they have that type of vibrations. But as we know, our sample is protein, and we can try to understand that that protein is having a amide bond, and amide bond will be having two types of vibrations. So we can predict it, but prediction cannot be as much as accurate what we are saying here.
You can use the FT1 to see like 1004 is the macromolecule for phenolide. You can use tryptophan, tyrosine. I think I saw yesterday. I'm coming to that. So these are complementary technologies. So if you use FTI, the molecules is definitely vibrating in carbon, nitrogen, hydrogen. So this is the composition of the protein molecule. So if you probe that with FTI, then you have to cross-check it that it's a pure system, but it's a heterogeneous system where a mixture of things. It is mostly shocking movement of the molecules that generate this the peak. And then coming to transmittance and observance, you just have to click a button that can convert to transmittance mode to observance. And so the same thing, it doesn't change the structure whether it's transmittance. Yes.
I think we use the linear regression models we can validate for FPI. There is a linear regression model. So with the help of that, we can manage FPI values whatever. Because graph we can take either the province in and convert the things we can convert to transistors also.
Yeah.
And so one more query I have. Beta
Yeah.
beta when there is something beta
This is 1662.
huh?
1662. Then the
What is that? I'm asking.
okay. Sorry. So like in the second structure, you have alpha helix, beta sheet. Within the beta sheet, you can have beta tone, beta sheet. This is within the beta structure. So it's a secondary structure of the protein. Yes.
See, there are three structures.
Yeah.
There is a helix, sheets, and you can say turns. There are reverse turns and these small parts. Why? And sheets and
random points. So denature करें किसी beta sheet करें क्या एक्टिवेशन
beta sheets and alpha helix. When we talk about it, one is basically generated because of the alpha helix structure, and sheets are simply because that we have two polypeptides, so they have an antibody and generate the sheets. And beta turn is totally different type of structure. It's neither coil as a proper model sheet, but it's a type of secondary structure. So it's a third category of the secondary structure.
So by full denature
No, it's generally in the native protein they have see the
Okay. Come here. This is this direction. This is the plane, XY plane. If this is beta sheet, if I look like this direction, this is called antiparallel beta sheet. This goes this direction, this goes this direction. Alzheimer's data, this is like this. We are talking about. And if you have random coil is denatured protein. This goes like this. It's a coil. So helix is the most stable structure because within the beta sheet is also stable structure, native configuration, and some protein have random coil.
basically it is generated because of the different type of hydrogen. We generally say N plus N4 leads to the helix. But if you have N plus N2 and N plus N5 type of interactions, then it generates another structure that is called turns.
Okay, I was putting once few slides and explaining the structural detail. I thought I presume that we are clear about all the structures. So
like one and one seat okay. Then it will fold upon itself. This is turn, and then N to C. This can be parallel, and N to C, every turn is N to C. Anti-parallel, one is N to C, other one is C to N in this direction, reverse. So these are the single polypeptide chain is folded upon itself. It will take turns at the terminal ends. That means these are like N to C. Yeah, native structure.
thank you. Okay. Let's see the FTI spectra of this molecule, how moisture is affecting it. We have different band region here. So this way is stretching. Amide 1 region here, amide 2 region here. So 4.6 to 17% moisture content. You see, if we are increasing the moisture, the intensity of all the three major regions are increasing. Means the molecule, the conformation is changing. Okay, we are just adding water from different moisture. The conformation is changing. Now, please look inset here. Inset means So this is the amide 1 region. This is the 1600 to 1700 region. This is the observance here. So if you look at this midpoint of this peak, this is 17% moisture, and this all up to until 13% moisture. So this is 1634. The position of peak means the FDR data is telling the molecule predominantly in the beta sheet until 13% moisture. Then when your moisture content is increasing, it's no longer at beta sheet. The secondary structure change to beta sheet. There's a transition occurring at that moisture to beta strand. So means how glass transition temperature is DSC is telling how this data is related, this structural data is related to DSC data. I'll come to this later, then I'll come here. Please look at the table here. Directly look at 13. The glass transition temperature is 18.5. Okay, means it's nearly at room temperature, so we can measure it. When adding 30% moisture, we do not detect the glass transition temperature. When the moisture is decreasing, the glass transition temperature is also decreasing. 4.6 moisture, 94. Now you look at this. So 11 until 11.4, that's the 29.3. That's the room temperature. Because this structure is changing from beta C to beta strand, means that the structural realignment is occurring, transition is taking place at that moisture. So please remember this 13% moisture here. So I think this is a take-home message here. If you are dealing with any powder, if the moisture content is above the 11-12% moisture, means your conformation of that protein is different. Okay, means it's no longer the genetic configuration. That's a clear answer. So that's experiment 2. So the first scan is the stress relaxation piece. This is the we have already discussed. Second scan we are setting the glass transition temperature. In this picture, the denaturation temperature is decreasing with the increase in moisture content, 26%, and the glass transition temperature is also decreasing, being plasticized. So what is the phenomena is happening here? There is a glass to rubbery transition taking place at moisture, 13% moisture. It's no longer in a glass. That is called the rubbery state now, because 13% moisture is affecting the molecule in such a way it lost its glassy nature. So it's a rubber now. Okay. That is called glass to rubber transition. So we go glass to rubber.
No.
Yeah.
I was very simple. Sorry, I'm failed to understand how to report that glass transition temperature from this particular curve. Because you have put some arrows here, even in the prior slides. But basically, are we looking at the sample at the time of this calibration, sorry, this calorimetry analysis, or by using this particular curve, we are identifying the transition temperature?
Okay. This is from here. This is the second scan data. We measured the glass transition temperature. You can I'll integrate this. This is delta Cp change, the heat flow change. That's the glass transition.
So you check the delta Cp.
This is the second order changes.
And then you have some threshold limit blocks in that.
Yeah.
And then you say that.
Yeah. This is integrated. You can see the slope here. Please look at here. By reading I, you can see your glass transition. I'll put a hypothetical line here. It goes straight like this. And another point here. The mid inflection point is on the glass transition temperature. Okay. And onset temperature with the year starts the reaction. And when it ends, that the end. The midpoint of the inflection point is known as the glass transition.
So basically, you take two time points, data points, and then try to compare throughout the scans. Which at what point my delta Ct crosses my threshold. As soon as it crosses, then I put that particular glass.
Yeah, glass transition. Usually midpoint of this is the glass transition, the inflection point. Now this clear now, everybody? Okay, next case. We have discussed this glass transition temperature. The T mass is decreasing and increase that, and the denaturation temperature is decreasing, Tg is also decreasing. So these are very.
So what is the difference and what is the meaning on-site temperature and the glass position is coming, the air temperature is coming? If we will do on the equipment, it will show us on-site glass position and air temperature. I mean, what is the meaning of that? Even if it is
Okay. It's a very good question. So when you heat it, the sample, the reaction will take, because it's a very large window. Glass transition is very difficult to measure. It does not happen in a narrow window or 1 degree or 2 degree. You will take a pinpoint test for glass transition. No. It's happening in 20, 30 degree range. So we are integrating 20 to 30 degree temperature. So it has to start from some region and end at some region. And midpoint is the glass transition temperature. Where it starts, that's called on-site temperature. Where it ends, that's called the air temperature.
Okay, for example, we have given the range between 20 to 40 degrees Celsius.
Yeah,
So
20.
on-site temperature would be?
It starts from 20, the start from 20 is 20.
So
And it's at 40.
the range we have given 20 degrees, so it
Yeah,
is
yeah,
on-site.
yeah.
So between the glass condition would come,
Yes.
and it will be 40 degrees.
Yeah. 20, suppose middle will be 30. 30 will be our glass transition and point is 40. This is the challenge actually. Measuring glass transition is a very broad transition. The difficult is that you want triplicate analysis, then where the baseline is there, and how you measure that one, inflection point, midpoint. So you start with the glass transition temperature.
Is it necessary that it will come in between only?
Yeah, it's always the midpoint of the inflection. Always in the middle. Yeah, always. Okay. So you have seen that the parameters of the plasticizing effect of the water molecule, and then how the glass to rubber transition is taking place. Coming to major bands in food components. So food will be basically major is protein, starch, and fat. We all know that. Major any chemistry defined definition of food will be that when we have surfactants, we have emulsifiers, water, and all molecules are there. So it's totally molecule. So carbohydrates and water. So these are the assignment. To refer quickly, that these are the bands that we will be looking when we take the spectra. Will clearly give us where it is vibrating, and the assignments are given here. So suppose you are looking for water here. So we say that this is within the limit of the amide band. It corresponds the protein band also. So we'll get confused with the water molecule. And the signal to noise ratio is a very hydrated system. There won't be any peak because it's most like water. So what you do in the practical solution, you have to increase the concentration of the protein. Basically, 10 to 14-15% protein is required. Very beginning, I thought if we can afford more protein, then we can employ this kind of spectroscopy. But if it's very expensive protein, we cannot go below microgram. We have to go for the circular nitrogen kind of study. You have to decide how much protein you can spend in this kind of experiment. So these are you can refer this the frequency here. So this is the assignment of band. This is beta sheet and beta strand, alpha helix is here, turns and type. So suppose you have any protein measure, you are interested to characterize it. You will be looking the signature spectra, whether it's amide 1, amide 2, or amide 3. Then when you're going to amide 1, then you do a deconvolution. After the deconvolution is algorithm basically. Software will tell you have to select your own parameter. So when you go through the algorithm, then it will give the buried component. Then you assign those peaks that I shown in the before. So these are the peaks. Then you can say that this protein is predominantly beta sheet or random or alpha. Simple way to take a denatured protein, you just heat denature it. Means you know its denaturation temperature. Any protein you take, put in 100 degrees and for 10 minutes protein is denatured. Okay, then maybe whey protein you take, I'm talking about 10 minimum 10 minutes. Then you take a native protein, then study in the microscope under the FDR spectra. Then you will definitely see how the peak shift is occurring. This is experimental detail. Quickly you can do yourself. Just because in a hydrated system you do, and also do in a powder system. How the denatured powder system is, you can means you have to have suspend in the water system, then denature it, or you have to heat in a DSC, then start it in this one. That way you have to design the experiment.
So one more doubt.
Yeah.
So this selected digestion bands are there. So in that there are two frequencies. One is the 3600 band and the second is 1650 for water. If water is present in that two component or not, possible you are using. So if say 3200 to 3600, the range is somewhere in between. That means it might be containing water into it. Am I right? So sir, how will you confirm that? I mean, along with the 3600 band, 1650 band is coming, then you will confirm that water is present.
Okay.
Is it so easy?
Usually what you people do, when design any buffering system in your FDR study, water interferes with that. Yesterday I was mentioning deuterated water you use D2 instead of water here. Instead of HCl you use DCl, and instead of sodium hydroxide you use NOD. Okay. So in case of pH it's known as PD. People don't, because you have replaced water with heavy water. The water. So water does not mask your peak. So you have eliminated the water part. That way you conduct your study. But if you have to design the experiment such mostly deuterated water you use, so it doesn't interfere at all. So you are starting the molecule as such. Okay.
Means we are not impairing, we are just studying.
Yeah. It means
Interference would be done by further what study? Using what?
if you are interested to look into the question is for further you want to use
Okay, for example, I have done studies on cellulose nanocrystals. So I have taken the Fitbot settings, one is then nanocellulose and nanocellulose crystals. Okay, so bands would be coming. I mean, band are coming. Carbon oxygen stretching is there. The OH stretching is there. So how would I come to learning what further study I can do? For the interest for that I'm asking.
Oh, okay. So means suppose you have assigned the peak, so you have done any modification to that, or you, like cellulose extraction, suppose lignocellulose biomass conversion people are using, use fungal enzyme to convert it to that, or you have a blood combination of enzyme, then you see how much hydrolytic enzymes are active in that, and then how much modification has taken place. Then you take the those material and then look at the on this in the spectra. Then you will see how this pattern is changing in a time course, how the structure is changing. All these things you can do. This is the next step. You should have some of the effect in temperature, pressure, enzyme use. So that way you can design. Same thing, same principle.
Actually, I said exactly the same thing also. So using that also we can.
Yeah, you can. And all the techniques are complementary. It's up to you what you are employing here. So coming to what's happening here, the recombinant spectrum means if you look at the insect here, so I was talking about this peak, 1600 peak here. So let's call 1634 peaks. So you can see beta seed is converted, changing to anti-parallel type of beta seed type of seed. Means that is the glass to rubber transition taking place there. And the rearrangement molecules, and then molecule no longer as a powder, that is called collapsing, means macroscopic collapse. So the agglomeration really takes place there. To summarize the detail of what is happening here, major intensity of major bands are changing with increase in moisture, or it's amido or stress means there's conformational change is occurring. And there is a glass to rubber transition taking place at moisture 13 molecular collapse is very higher, moisture content 26 degree, means this transitions really occur at 13% moisture content. That's last year of the transition.
Sir, can you please again explain what is deconvoluted statement?
Okay.
Is dissolving the graph is I don't know.
Yeah. Deconvolution means if you have the like 16 to 17 and we are studying the amide region, the mid peak I am showing here, you are only seeing here either 1634 or 1630 here. But we do not know other components of that spectra inside that. It might have random coil, antiparallel beta seed and other things. But the major peak is the beta seed or beta strand here. So we have to resolve the buried peak inside. So we are using an algorithm here to disintegrate the peaks. So it's like a seizure. So we are using spectral algorithm to deconvolute. That's called deconvolution. Without affecting any, you are not introducing any artifact to the system. In the software you are doing. So now you can see this is the deconvoluted spectra here. So you can see this is no longer a single peak here. You have 1, 2, 3, 6, 7 peaks in the job. So here. So you can see this first and the black one is the top one is 1634. Now this is the deconvoluted spectra. This is the deconvoluted spectra. So you have resolved the other peaks. So now you know what are the other components. When you heat it, the components will change. But buried peak will the only single spectra will shift from one wave number to other wave number.
Means in this we are taking test is also random plus.
Yeah.
But while we are seeing the result, it is in the
Beta seed. Yeah. Okay, we're talking about denatured versus native protein. How is it happening? Let us have, we already know that at a 4.6%, we are comparing a native protein here. If you take 4.6 and compare already 13%, we know that's a rubber state protein. So we are taking glassy type material, it's 4.6, and then we denature the protein. What's happening here? Same moisture content also, because moisture has effect on the structure we studied now. So this should, when you compare denatured versus the native, you have to have same moisture. Otherwise, you will not see the effect. Please remember this. So if there is no same condition, so you cannot have the same result. You will be looking apple and orange, and the same protein is there. So here, the denatured protein, you can see the frequency is increasing, the intensity is increasing within same condition, but within the denatured protein. And the position of the native protein configuration is 1634, that's beta C. When you denature, it's an anti-parallel type of beta C. You can see 1623, no longer at the same position, like this, anti-parallel beta C. So this protein is a denatured protein with a different configuration. So now you can throw itself that the accepting of iron band. Is that clear? I will continue this when I come back after the break, couple of minutes here, but we still wait. It's not 11 now. But I'll just explain something, and then when I come back, we'll again continue this. Is that okay with you, all of you? Okay, so when you are doing any structural studies, certain things, because you will be getting confused otherwise. You are doing correctly, but you have not designed your experiment properly to have the comparison. Like moisture, you know. So if you have control conditions properly placed, then you get the exact comparable data. Same result, but you have meaning for that. So the real simple parameter. The lesson is not teaching, but just learning how we have done, so you can apply in your own field.
So, sir, it means that when we get a single peak, so it is that beta sheet is most prominent, and when we deconvolute, then we get to know what are the other components as well of the protein.
Yeah.
Any other query regarding this,
or we will address it in the tutorial session, or once we resume the session at 12. Right? Sorry, 11:30. Okay.
We resume the session post teaser. Now I invite you all for the session.